The Center for Systems Genomics, so if anybody's interested in that, please come and ask me. If you've got any questions, there'll be a little bit of question time at the end, but I'm around for today and tomorrow and then leaving Wednesday morning, and so if anyone want to ask any more. I'm, as you've heard, a statistical geneticist, grew up in maths mainly, maths and stats, and then I've been working in genetics most of my professional life. And what I'm going to tell you about today is some new ideas about heritability. And now most of you have probably heard something about heritability. Everyone's heard a little bit about these things about heritability of height and IQ, and it's quite controversial in some ways. And so I want to go over that a little bit about what's wrong with the idea of heritability, but there's been a lot of new thinking about it, and there's a whole new school of thought that's really kind of propelled the field of complex disease genetics and understanding the architecture of genetic architecture of traits quite a lot since 2010, when this paper came out from a group in Brisbane, and it was sort of obvious in some ways, like many good ideas, but also revolutionary in other ways. And I want to talk you through some of that. This was the exciting plot in that paper that revolutionized the world. There's a whole lot of points there on a line. Is that line flat, do you think? It looks pretty flat, but it actually has a non-zero slope, and that's the exciting part, because the x-axis measuring some notion of relatedness between individuals, and the y-axis is the squared difference in phenotypes between those pairs of individuals. And so the idea is that the more related they are, the more similar their genotype, their phenotype, and so the squared difference will be a little bit less. That has a negative slope. That's very exciting. You might not think so, but I'll try and explain why in a little while. Actually, that regressing against the squared difference is not the technology that's actually used for the estimation, but it's the easiest way to think about the problem. Right, so just go over some basic ideas. Heritability is some kind of measure of how genetic a trait is. As I said, it's kind of controversial. It's much misunderstood. People often say, oh, you know, the heritability of this or that trait is so much behavioral or IQ or something or so much, and think they've made a statement about the nature, but broadly speaking they haven't, because it's very environment specific, and that's where all the controversy really comes from. But we won't go into too much about there. I'm mostly going to be talking about narrow sense heritability, which means additive contributions to the phenotype, and so we're trying to work out pretty much how much of the phenotypic variance can be explained by additive genetic models. So the underlying idea is just a simple linear regression, like I've shown there, and the Y is some phenotype that you're interested in. I'm going to be switching between binary and continuous phenotypes. There are some issues there that I'm just going to fudge over. You can proceed as if a binary variable were continuous, and that's legit to a certain extent. I won't go into all the details. But the problem with this, linear regression is very simple. The problem is that it's a regression on the causal variance, and the beta coefficients are the effect sizes of the causal variance. But if we knew what the causal variants were, then we could go home and close down most of the genetics departments, because that's what we're trying to find, and so this equation is not directly useful, in terms of just going down and working with it. But of course it's a framework for thinking. There's various ways of trying to get at this. There's a lot of ways of measuring heritability that are based on known family structures with people of lots of pairs of individuals of a fixed relatedness. But the most flexible framework, and probably the most widely used, is this mixed model framework. Now, this is another exciting equation, but it's also looks pretty trivial because it's just the combining two numbers here. Gamma and epsilon are just numbers, the genetic sort of liability, if you like, and the environmental one. And so the interesting part of the equation are the variance structures, because we don't know either of those things, but we think we know something about the variances, and that's the key to being able to estimate things. So we sort of assume in this simple model you can have more complex structures, but in this kind of simplest setting we assume that the environmental contributions are independent across individuals. That's what that's saying, with some variance sigma squared e. But the genetic contributions are not in general independent. They're correlated according to relatedness. And so that's the kind of key idea here, that this term has a correlation structure that reflects relatedness, and this has independent correlations. You can estimate these parameters. The kind of standard technology now is using REML. If you just leave out the RE, the ML stands for maximum likelihood, so it's in a sense roughly straightforward maximum likelihood estimation. REML means restricted or residual, or nobody's quite decided what that, but it's a way of dealing with the nuisance parameters that tries to reduce the bias of, because maximum likelihood estimators are typically biased when there are nuisance parameters, and here we've got lots of nuisance parameters, and so the bias would be a substantial issue. And REML, if there's a sort of Bayesian way of thinking about it, is that REML is integrating out the nuisance parameters, or corresponds to integrating out the nuisance parameters with respect to a uniform prior, which is generally improper. But in any case, there's software to do this now. There's been a lot of developments in the last decade or so to do it sort of bigger and bigger and faster, and so that is not so much a problem anymore. But again, the variance structure that we really want involves the true causal variance, which we don't know. And so what we've traditionally done instead is approximated this variance structure by kinship coefficients estimated from pedigrees. So the idea is that if you and I have a certain relatedness, then that gives us some expectation about how many alleles we share through common ancestors as a result of that relatedness, and that will do as a proxy for how many causal alleles we share, and therefore the correlation structure due to excess sharing of causal alleles due to relatedness. Well, and I've been doing a little work thinking about this, a little bit of an aside here, that sort of rather bizarrely, people then came to think about the pedigree relationships as being really sort of fundamental and central. So one of my messages is that they're not. The pedigree relationship is firstly, has a lot of weaknesses, and it's not what we want at all. It was always just a proxy. It sort of struck me coming into this field as an outsider how everybody's, all the textbooks were full of, standard population genetics textbooks would have something about kinship coefficients, and it was all written down as if this were facts and unproblematic, but it's riddled with problems. There's no definitive pedigree, right? You can always add more ancestors and change the kinship coefficients, and there's no right answer. You can't, if you keep adding ancestors forever, all your kinship coefficients go to 1, which makes them useless. The other thing is that I think people sort of lost sight of the idea that what we really care about is how much genome you share, and these pedigree coefficients are only getting at some kind of expectation. More generally, there's actually no, I sort of found this a little bit shocking considering all the sort of textbooks just treat this as an issue of which there is no problem. But the answer to is, how closely am I related to you? This question doesn't really have a well-defined answer. We don't nowadays, we used to, even if you say, so-and-so's my aunt or my cousin, you think that's kind of straightforward, but actually it's not, because that's only telling you about one lineage connecting you with that person, but there are many other lineages, and so to fully specify the relatedness, you'd have to say something about all those lineages, and now with genomic scale data, we can actually start thinking about those more subtle differences, and the whole traditional concept of relatedness and kinship just really falls down. It doesn't work anymore, and We tried to grapple with these ideas and recently wrote a Nature Review Genetics paper on it going through some of these issues. And it's a little bit aside to my main point here, but this issue of the variability in allele sharing for relatives. Now, this is in the artificial scenario that most people assume that siblings are only related through one lineage and the other lineages are all infinite in length. That's a kind of standard assumption that I was just criticizing. But it just helps remind you with these here that even under that kind of optimistic assumption, there's a fair bit of variance. I think these are 95% intervals in the amount of genome sharing between siblings, for example, and then all the other relatives. And siblings are really unique in this list in that they are kind of clear and distinct from the others. All the other kinds of relationships, the confidence intervals overlap. And so just on this allele sharing, you can't distinguish relatedness. Also, a thing that I thought was kind of interesting, there's a lot of information on this table here, but the one thing I just wanted to highlight is the probability that you share anything at all from a common ancestor is 1 for close relatedness for a close common ancestor. But once you get to half-second cousins, so that's 3 generations up and 3 generations down, you start to have a non-negligible probability that you actually don't share any DNA at all. And so you can actually think about the scenario of a person with 2 great-grandchildren and they share DNA with the 2 great-grandchildren almost certainly, but there's that 0.5% chance that the 2 great-grandchildren are genetically unrelated. In fact, that they didn't actually receive any DNA in common from that ancestor. And I just thought that was an interesting idea, but it's sort of just emphasizing this idea that what matters is not, so that half-second cousin relationship would then be kind of useless because they don't, although that gives you an expectation for sharing some of the genome, there gets to be this probability, still small then, but it very quickly gets bigger. And so it's kind of useful to keep in mind that once you go back more than about 300 years, if any of you are interested in this genealogy, that can be a fun thing to do to try and trace back your ancestors, but you probably didn't inherit any DNA from them unless they're beyond about 300 years. And so if you're descended from William Shakespeare, for example, and you've got a paper record proving, then that might be very exciting in some way, but you almost certainly didn't get any DNA from him, and so the relationship is, in a genetic sense, is sort of meaningless. So, right. So coming back now, that's what's wrong with the old-fashioned pedigree stuff. It's not to say that it doesn't work at all, of course. It makes a reasonable approximation. The plant and animal breeders have been using it successfully for years, but nowadays in genome scale data we can start seeing its deficiencies. And now we can think about actually directly measuring the genome sharing through excess sharing of SNPs. So that is clearly better in a sense, but it brings with it its own set of problems because there's lots of ways you can measure relatedness. As I already said, not only are pedigrees problematic, but even if I gave you the genomes of everyone in this room, and also I gave you the genomes of all of our ancestors and then said work out the relatedness between all the individuals in this room, the question is ill-defined. We don't really have any meaningful definition of what it is we're trying to calculate. And in reality, when you've got SNPs, you've got extra problems here because there's quite a bit of confusion about IBD, people, identity by descent, sharing genomes identical from an ancestor. It sort of means slightly different things in different concepts, because you talk about having half your genome shared IBD with your sibling, which makes sense in a certain sense, but on the other hand, you share 99.9% of your genome IBD with everyone else, every individual in the room. That's because the 99.9% is back to any ancestor, whereas the half with your sibling is only counting the shared inheritance from the parent. That's why there's that discrepancy, but it's just another reason why there's much confusion about these points. And a whole lot of papers, when they report relatedness coefficients between individuals, I always find this frustrating because they don't really say what they mean, and there are lots of different definitions, and it's kind of remarkable that few people seem to notice this, and people are computing relatedness in different ways. But this is quite problematic that with SNP data, the answer you get for any relatedness coefficient you might want to think of depends often quite sensitively on the minor allele frequencies of the SNPs that you happen to choose, and that depends on the SNP chip and also on the QC that you implement, because getting rid of the rare alleles, which is a common thing to do, has a big impact on these measures. So that's another one of the sort of problems along the line. So I won't go into any great detail about here, but there's lots of different ways you can think about measuring relatedness. What most people do is some kind of, you come up with your favorite measure at a single SNP and then just average it over the genome. But of course that's not the only way to do things. You can start thinking about chunks of DNA that are shared and trying to measure them. But that gets problematic because, you know, we're all sharing a huge number of small chunks of DNA, and we can't really detect that sharing very accurately. And what people do when they talk about detecting IBD between individuals, they have something in their mind about an idea that they're looking for big chunks of DNA, but there's no real good definition usually of what big means. And so that's quite confusing. There's a bit of controversy. There's a Oxford group, or they were based in Oxford, with Simon Myers and a few others. The name's just slipping me, but who were sort of arguing that this was a much better approach using certain methods that they have developed. But actually for many, and we've looked into this, and for many purposes that I care about, this approach always works much better, and this is what, but maybe longer term this might be a better approach. But I just want to highlight again that you'd sort of think that these basic measures of kinship and relatedness was all sorted out in the textbook decades ago, and it's quite bizarre that it's not. And one of the little problems is if you've got two SNP genotypes, how do you measure the similarity between them? Well, a standard scaling is this one, that if you're both homozygote for one allele or both homozygote for the other allele, that's a perfect match, so you get 1. If you're homozygote for opposing alleles, you've got nothing in common, so that's a 0, and all these others are sharing a score a half. And the logic behind that is that if you choose one allele from the first genotype and one allele from the second genotype, what's the probability that they're the same? That's what those numbers represent. But there's a bit of controversy about matching heterozygotes, whether that should be counted as a perfect match or not, because there's one argument: if you're matching homozygotes, you've got the same genotype. Matching heterozygotes, you've got the same genotype and it's a perfect match. But the other argument is they might be out of phase, in which case it's a perfect mismatch. So classically, that was scored as a half, as a compromise between those two. But if you know anything about sort of GWASy kind of human genetics, the software Plink just dominates everything, and whatever Plink does has now become correct, and it does something different. It scores matching heterozygotes as 1 rather than a half. And so lots of people are using these different formulas and coming up with quite different numbers. The upper bound on those is quite different, and hardly anyone noticed the difference, which is kind of bizarre. But actually that's not what most people used now. Most people are using this average allelic correlation. So that is, you take the genotype scored as 0, 1, 2, take off the mean, and then that's for the first individual, and then do the same thing for the second individual, and work out what looks like an n equals 1 estimator of the correlation coefficient. And because it's n equals 1, that's a really lousy estimator of a correlation coefficient. But you can argue that it's the same coefficient at every SNP on the genome, which is true under certain assumptions. And then if you average over the genome, you average a huge number of lousy estimators, and you get a pretty good value. Although, actually, I've slipped up there. You have to be careful if you're talking to me, not to call these things estimators, because I've already said that there's no it's not obvious what is the parameter that they're estimating. So they're not really estimators, they're just numbers. They're just measures of something. So as I've said here, this is better than the pedigree-based because it's based on actual allele sharing. But there's a problem now that we've introduced, that the causal variants only contribute if they are tagged by the SNPs. And so that creates a bias towards common causal variants, which are better tagged than rare causal variants. So you might think that makes this kind of whole approach as hopeless for measuring something like heritability. And in a sense it is. And again, there's a lot of misunderstanding here and a lot of nonsense that's said, because people say that this only measures the common variation. Well, it doesn't exactly. It does tag some of the rare variation and most of the common variation. But I'm going to argue that it's still useful despite the wooliness at this stage. The total numbers are not really very meaningful, except as maybe some bounds, but the relative values can be meaningful. So I started off my talk with the paper that I said was kind of revolutionary. This picture sort of shows a little bit about why the method was a bit sort of surprising and controversial. So basically, on the left panel, we've got a lot of people of greatly differing relatedness going up here and fitting a regression line. As I said, this regression line is telling you about heritability in some sense. Over there, we've got rid of all the close relatives, and we only include individuals in this narrow range. Now, what do you think is going to be the better fitting regression? Which line would you trust, the one on the left or the one on the right? Anyone got any views? Sorry? Well, I think the superficial answer might be that you've got a lot more information here over a wider range of the scale. Here you're getting information only on a very limited range, and so you might think you're getting a better estimator over here, and you are. But the sort of radical idea of that Yang et al. Paper was to use the regression on the left and only use the unrelated individuals. So getting rid of the relateds throws out a massive amount of information, which seems mad, but there is an argument for it. Why do you think we want to throw out all this valuable information? Anybody got any guesses? No, it's early on Monday morning. That's good. The problem with this is it's very variable over samples about the degree of relatedness you've got, and the answer is going to depend a lot on how many relateds you happen to have in your sample, and it's probably not going to reflect population patterns of relatedness. Over here, if you're only using the distantly related individuals, that's very sort of repeatable from one sample to the other, because if you do the same process of taking out close relatives in a different sample, the information you get is from sharing small chunks of DNA from remote ancestors, and that's quite reasonably consistent over estimators. So this is why this was the kind of innovative step in a way that the Yang et al. People did, was to throw away the relatives, and you've still got enough information here because the vast majority of your points are here for many samples that closely related individuals are relatively rare in a population sample, so that's the advantage with this approach of only using unrelateds. So the approach has been thrown at a lot of different problems, and here this is just representing some of the heritabilities that have been estimated. The SNPs that have been significantly detected from GWAS represent in general only relatively small amounts of heritability, although growing. This pinkish bit is bigger now than it would have been a couple of years ago. This is the heritability that's been attributable to other SNPs using this Yang et al. Approach of using SNPs to measure relatedness with this allelic correlation, and then trying to say how much of the phenotypic variance can be explained by that. And this light blue part is also getting bigger with better and better imputation and eating into this dark blue part, which is the other contributions to additive heritability. There's been a lot of discussion about the meaning of this thing, that the light blue represents common causal variance and the dark blue represents rare causal variance, but I've just been saying that isn't true, and more and more as you're getting better imputation and so on, you're picking up more of the rare causal variation and explaining more with this SNP-based heritability measure. And of course now we can start thinking about full sequence data, which we don't have in big scale yet, but we soon will, and thinking about, we should be able to explain nearly all of the heritability. Now, there's a paper just come out which does try to approach the full information from sequence data, and they found that they could explain much more of the heritability. It still left a gap, but they're also arguing that the total columns here are the traditional heritability estimates, which might be wrong for a number of reasons. There are several kind of arguments why the traditional heritability estimates might have been overestimated. So we had a look at this mixed model estimation of SNP-based heritability and found that it was pretty robust. There are problems with sensitivity to population structure, but there are various ways you can check and adjust for that. But we did find that it was very sensitive to linkage disequilibrium. So that's the correlation between neighboring SNPs. And it's kind of easy to understand what's going on here, that if you have high region of LD, a lot of correlation among the SNPs, then there's multiple tagging of the same causal variant, which then gets overcounted in the regression. And in low LD regions, it gets undercounted. And so this can lead to a bias or an inaccuracy. So then Doug and I developed this scheme of reweighting to adjust for linkage disequilibrium. This actually had some interesting kind of consequences. So the idea here is that if you have 4 SNPs that are all very close together and they're all in very high LD, then they should all get about a quarter weight, because essentially it's one unit. Now, we thought this was pretty straightforward and uncontroversial, but the Brisbane lot who introduced the method were not very happy about this idea, which seemed to sort of be critical of there, which was achievement. So first of all, here's that our method worked in our simulations. It corrected for this wrong estimation. So the green and the red lines represent 80% heritability in one simulation and 50% in another. And in our simulation, of course, it all works beautifully, but it's a little bit more complicated than that. And with the paper that just came out from the Brisbane group, we're showing another problem with 80% heritability and showing our method to be overestimating dramatically. Well, this only just came out a few weeks ago, so we've only just seen it. And we're very annoyed about this, that's for misunderstanding the point of the method, but it kind of raises an interesting question. So this is their Yang et al. Again with their 2015 paper and saying that we were overestimating. But it all comes down to what simulations you choose to do, as is often the case. And their simulations distributed heritability across SNPs and sort of uniformly with respect to linkage disequilibrium, i.e. they didn't pay any attention to linkage disequilibrium. There is a function of minor allele function. So our method is based on the idea of downweighting apparent contributions to heritability when linkaged in regions of high linkage disequilibrium. And back here, I think it's pretty clear why that might be a sensible idea, because you would understand that, that these 4 SNPs close together, if they're tagging anything, they might all be tagging the same thing. But it's less clear when you get down to the level of sequence data. Nevertheless, it becomes an interesting empirical question that we don't have any good answer to, that if you have a group of SNPs all in very high LD, let's say there are 5 of them, and you have one SNP over here, how much causal variation are the 5 SNPs in high LD going to tag? The assumption underlying the simulations of the Brisbane group is that 5 SNPs is always 5 times as much heritability. And it seems to us obvious that that's wrong, but it's not completely clear what the truth is. To some extent, it's an empirical question, which now becomes a kind of interesting one to check, which we want to go on and have a look at and prove that their criticism of our paper was silly and they're wrong. But it's an interesting thing to look at. Anyway, back to the story. All of this has opened up an interesting way, new way of thinking about heritability, and really the traditional mixed model approach, which was clung onto for quite a while. Most of this, the Yang et al. Paper, are still working in this mixed model framework with REML, but actually it's now become completely unnecessary. You can, instead of the REML model is everything, all of this, there's always a Gaussian assumption that I haven't assumed here. Everything is assumed to be Gaussian in the likelihoods in order to maximize them and apply REML. And it turns out that that mixed model formulation, it's not completely obvious, is completely equivalent to something like ridge regression, as a kind of a genome-wide regression with all the SNPs in the genome included, but shrunk with a kind of Gaussian prior, if you like, in the ridge regression way. And this is kind of interesting way of looking about it, and whether this way of representing it or the traditional mixed model way, which one is computationally better, it depends a little bit on the subtleties of the problem. But certainly the genome-wide regression on all the SNPs now. Remember, I had a simple regression model before where the unknowns were causal variants. So this now formally looks the same, except we're using the genotype, all the genotype SNPs as proxies for the causal variants, and then because it's a regression with a vast number of predictors, you've got to do something about the overfitting, and so the sort of default thing to do that's equivalent to the mixed model analysis is a Gaussian shrinkage like ridge regression. But of course, it opens up the way of doing other kinds of shrinkage. The Gaussian is not necessarily a particularly good one, and there are other possibilities available which people are interested in. But this is the really interesting part, is that you can now think about, which is not very natural in the mixed model framework, but in this genome-wide regression framework, it's very natural to think about using this kind of technology for just certain parts of the genome. And so you can start thinking about the heritability attributable to certain genomic regions and functional classes of variants, and that turns out to be, I think, quite helpful. So again, there's a lot of Yang et al. Papers I'm referring to. I think this is the third different one. They introduced this and did genome partitioning, as they called it, and genomic partitioning. But we've been playing around and doing some interesting things in this area as well. So when you look at the heritability of a region, you naturally sort of want to compare this with something like a kind of default or background level. And so the thing that we thought to do is to compare the amount of heritability explained with some measure of the total genetic variance of a region. So that's concept. The heritability explained as a function of the genetic variation is, we've called this intensity of heritability, and looked at things like, in this picture, this is the intensity of heritability in exons for the 7 traits in the Wellcome Trust CCC study, and then looked at that in intronic and in flanking regions of the exons, going out 10 kb and then 20 kb. And so we found quite a bit of all of these things. There's quite a bit of differences across the traits, but if you take some kind of average, by the time you get 40 kb away from the exons, then you're down to just kind of background levels, more or less, of heritability. But the other interesting thing is that, broadly speaking, for many traits, there isn't very much intensity. So the heritability explained by exons is only round about background level. It's just a bit higher for some of these traits that have a big MHC component, which we thought was kind of interesting, but we went on. So here's the intensity of heritability in exons. So again, thinking of what we're doing here is just looking at how much of the variance can be explained by SNPs in exons using this Gaussian shrinkage regression kind of idea. So the technology is pretty simple, and anybody could have done this 10 years ago, but it wasn't really, people weren't really thinking about the problem that way, and it was going via this route of thinking about heritability that led us into here. And so although this idea is not, in broad terms, new, I think we were the first to kind of measure it in some kind of precise way. And the relative, so this is some measure of how much heritability is in the exon relative to a background level. And you can see for most of these traits, it's barely elevated at all. It's just a bit high for type 1 diabetes. This intergenic means far from exons. In this case, I think more than 500 kb or something. So these two together don't make up the whole genome. But then if you're just testing for significant difference between these two rates, it is significant for some traits and not for others, which we thought was also quite interesting. But And we can also look at the intensity of heritability for eQTLs and see how much, if you've got your favorite set of eQTLs from some particular study or some particular tissue, how much of the heritability of different traits is explained by that. And that, of course, is going to depend on what tissue your eQTLs were determined in. So this was from one particular breast cancer study that Doug helped a bit with, and we found a huge proportion of the variance was explained by eQTLs for these two autoimmune diseases, but not for the others. And the pattern kind of held out to some extent, but not very closely in comparison over different tissue in which the gene expression was measured. So I won't go, I mean, there's a lot of information here. I won't go into any details, just to kind of broadly talk about the sort of ideas of what you can do when you start thinking about measuring the heritability in different parts of the genome. You can also, so heritability now, I guess the point I'm saying becomes, or at least some variance explained related to the idea of heritability becomes a kind of common currency to explain things. And you can start looking at the correlation of the genetic component within individuals across different traits. So this needs a little bit of explaining. These numbers are minus log 10 p-values when the histogram bar is significant. And you'll notice, so this is SNPs that were significant for schizophrenia on this row, and then how significant are they for these other orders, and we found that they were significant for bipolar disorder and for rheumatoid arthritis. Crohn's disease SNPs were highly significant for Crohn's disease, which might seem obvious, but just as a sort of test here, we had two completely different studies of Crohn's disease, and then they're highly significant, as you would expect, but also for rheumatoid arthritis. And this is just, again, I won't go into details, just sort of playing around with things that you can do with this common currency of heritability. But the other thing that we've just been working on is these gene-based tests, where this component of variance that corresponds to to some extent to a traditional notion of heritability makes a good basis for a gene-based test, where you can look at, where you can test for significant association rather than of individual SNPs. We know for a long time there have been different ways of trying to combine SNPs into a single test of association with a phenotype, and we think that this approach, based on the heritability idea, works much better. Again, I'm just giving a sort of broad survey here, and I won't go into the details. There's some simulations here with 1 causal variant per gene, 2 causal variants per gene, 3 causal variants per gene, comparing with a whole lot of other methods that have been out there in the literature. And we find that so this GBAT was our method. I think this is all sort of in development. I think we've changed the name now. Using a kind of classical significance test and a Bayes factor approach, which on these diagrams are almost indistinguishable, but working quite well relative to the other methods. So another interesting idea, we've just applied it here to some of the CCC traits, and I just show you a bit of a snapshot. These diamonds are the significance level for the gene-based test, the classical one here with a p-value rather than a Bayes factor, but they're often pretty similar. And we don't think that completely replaces the single SNP tests. It depends on what the genetic architecture. If there is just a single variant in the gene and it's well tagged by a SNP, then a single SNP test is going to do better. But if there's multiple causal variants or poorly tagged, then the gene-based test can do better. And we've been applying this. Unfortunately, Doug and I are involved in an epilepsy consortium. Epilepsy is one of the most complex of complex diseases. It's not very highly heritable, and there are hardly any SNPs. But we have found, by going down below the gene level now into looking at exons and introns as separate entities as the unit for testing, we've been able to try and glean a little bit of information. So this is what we found with a meta-analysis Again, heritability is the common currency for combining information at a gene-based level across cohorts in a meta-analysis. So we don't think anybody's been able to do this before. Meta-analysis has always been done at the single SNP level, but we can combine the gene-based test with a meta-analysis. Unfortunately, we didn't find any good hits, but this classic, what's always been the strongest gene for epilepsy, did come up quite strongly, and there's been lots and lots of Mendelian variants for epilepsy found in this gene. It hadn't previously shown up strongly in the GWAS kind of results, but we did find the heritability is pretty well spread across the gene. So all of this is showing you the different individual exons and introns, their contributions to heritability, and they're pretty much here. The other two hits that we found, we're pretty sure are kind of spurious, because one or two cohorts, they just showed up in one or in some cases two cohorts, and so there was something odd going on in those cohorts, and you can see it was restricted to just one. So unfortunately, we think these two hits here are spurious. With the SCN1A, which is the top epilepsy gene, this is just showing you the individual Bayes factors now for different cohorts. This might seem a bit disappointing that a lot of the cohorts look flat, but I'm not showing you the sample sizes, which are quite different, and so we thought it was pretty encouraging that the strong signals are broadly consistent with sample sizes and positive in quite a number of different cohorts. And remember, because these are Bayes factors, these values near zero do mean neutral. They don't mean evidence against the hypothesis. And so all of these are just representing just very weak negative evidence close to zero. But, right, I'd better, and we can do a sort of meta-analysis kind of estimate of heritability. I'll skip on. Again, epilepsy is one of the, as I said, a very complex disease. This is just showing the amount of heritability explained by each chromosome. On the x-axis, rather than chromosome length, we've got a measure of total genetic variation on that chromosome, so it doesn't strictly respect order. 1, 2, 3 are in the right order, but 5 has more genetic variability in our samples than 4, and so on. This is just showing that heritability is pretty well spread across the genomes, reflecting again this idea that we've estimated at least thousands of causal variants for epilepsy, and a real kind of uphill struggle for being able to make progress with the genetics. But nevertheless, we think we can do with these kind of genome-wide shrinkage type estimators some prediction that I'll tell you about now. So that, as well as So the basis of the story is, I told you a little bit about classical ideas of heritability. We went on to the new SNP-based ones. There are various problems with this. They all depend on measures of relatedness, and there's lots of different possible measures of relatedness. But all of that was just by a little way of background. Then, if you accept this allelic correlation measure of relatedness, which, as I said, there's no real kind of, diktat from God that this is what we must use, but this seems to be what most people use for no good reason as far as I can tell, but to prefer it over other measures. But if you do that, then you've got this technology which is quite useful. As I said, the total heritability explained is a bit hard to interpret, but all these relative amounts allocated to different parts of the genome turns out to be quite useful. It's this basis for correlations, genetic correlations across traits, gene-based tests, gene-based meta-analysis. So there's a whole lot of useful new tools that comes from this way of thinking. And all of that structure is having an impact on thinking about prediction of traits. So, before I say any further, of course, everybody is aware that in an outbred species like humans and for complex traits, prediction is quite hard. And so many people just say, oh, it's a waste of time. And I'm not arguing in general about this being clinically useful, but there are situations where it's useful, and it's always a good thing to try and do better. Basically speaking, just picking someone from a population and predicting their risk of a complex disease is a waste of time. But there are lots of situations where, for example, drug prescription based on classical risk factors can be improved with genetic predictors. And in epilepsy, we've got a situation where with the first, when people have their first seizure, generally speaking, because there are certain risks of drug treatment and a pretty high probability that you'll never have a second seizure, you don't get any treatment after your first seizure. They just sit back and wait until you have a second seizure, and then they say, okay, you can have some drugs. Now this, of course, is kind of a practical thing to do, but it's clearly suboptimal, and those first seizure individuals have much elevated risk, and so prediction, even in a low heritability trait like epilepsy, can be somewhat helpful. So here was our way of thinking about this. I mean, one of the things I talked about and that other people have been mainly thinking about is, instead of using Gaussian shrinkage, what about something more clever? I've done some little work on this in the past as well, something more clever with thicker tails, mixture distribution. So basically thinking about the prior going into this. So ours is a little bit of a different approach, or in that instead of thinking about having a more flexible prior, but the same prior for every SNP in the genome, we've been looking at relaxing the shrinkage in some areas of the genome. So basically you've got to do a lot of shrinkage to deal with the overfitting problem. But the problem with shrinkage, of course, is that you're then biasing downwards your effect size estimates at the really causal variance. And so we thought about doing a kind of first pass to look at interesting parts of the genome, and then in a second attempt shrinking less in those areas. So the model is just like before, but there are now multiple random variants. So again, I've got switched back to talking in the mixed model language, with latent components here. But this is still, equivalent, or can, under many circumstances, it's equivalent to a genome-wide regression. And if you use these allelic correlations, then it's no problem to switch backwards and forwards between those two formulations. So here's one way of thinking about it. These are effect size estimates without any kind of shrinkage for a certain trait. BLUP applies a uniform shrinkage across the genome, and that's what happens in this example here. Our adaptive MultiBLUP here found, I think, 3 regions. It looked by eye, I can't remember, to be honest, but I can only see 3 here. There's one there, another one here, and another one there, where we relax the shrinkage, and the idea is to try and get, if it looks like there's something going on here, then let's have the full effect of that region. And we applied it to some a priori regions for Crohn's disease and got a little bit of improvement. I mean, don't worry about what these numbers actually are. And as I've sort of argued that, broadly speaking, this is not practically useful, but doing better is helpful, and in certain situations it can be useful. And for the CCC traits, we found that we were pretty much always better than the other methods we found floating around. There are lots of prediction methods out there, of course, and we haven't been able to uniformly. But fortunately, this is from Matthew Stevens' group in Chicago, and they did a pretty thorough comparison against many other methods, and we did better than them. So we just relied on their comparison with other methods. The other thing is the compute time, that our method inherits the computational advantages of BLUP and is pretty fast. Whereas these other methods, the stepwise regression, is very variable in how long it can take, and the Matthew Stevens methods is quite slow as well. And there's also another method come up since Bayes-R. There's lots of other methods out there, but we've done some comparisons against Bayes-R. And we can do pretty big sample size. There's more than 10,000 individuals, we think. And we think, given a reasonable amount of computing resources, we can probably go up to 50,000 individuals and a few million SNPs. That would take, maybe a couple of weeks on a multiprocessor machine. So it's within the range of thinking about it. Yes. So just to finish off with a little bit of what we were overall able to do with epilepsy. Using these kinds of heritability ideas. This study, actually that's slightly out of date now, that was in our 2014 paper, but we've got more data and improved this estimate now. But we got some estimate. As I said, the estimate of heritability in epilepsy is moderately low. From everything I've said to you today, you should keep in mind that heritability is sort of almost meaningless in a general sense because of all these difficulties, but nevertheless it's still a kind of term that in some loose sense people find interpretable. As I've said, very few significant SNPs for epilepsy, so what there were in the way of even borderline significant SNPs only explained about 4% of the variance, not very much. on the basis of this, we did some theoretical calculations to infer that there are probably thousands of causal variants. We were able to find an estimate that there was, well, pretty close to 50% for the common genetic basis of the two main subtypes of epilepsy. We just lumped them all into focal and non-focal. There are other ways of classifying epilepsy and many more subtypes. And although it's a very imprecise estimate, we could show that it was significantly different from both 0 and 100. So there was some meaningful difference between these two, genetic difference between these two traits. And we were able to show for the one seizure individuals some useful prediction that we think could soon be clinically useful. So I will stop there. Thank you for your attention, and happy to answer questions that